The story of genetics is a fascinating journey that begins in a humble garden where a monk named Gregor Mendel meticulously observed pea plants. Mendel's groundbreaking work laid the foundation for what we now understand as the laws of genetic inheritance. This essay delves into the fundamental principles of Mendelian genetics, exploring the predictability of genetic traits and the mechanisms of inheritance that contribute to the rich tapestry of biological diversity. Mendel's first gift to the world of genetics was his insight into the predictability of inheritance patterns. He discovered that certain traits followed specific patterns when passed from one generation to the next. This revelation was encapsulated in what we now know as Mendel's laws of inheritance, comprising the law of segregation and the law of independent assortment. The law of segregation states that during the formation of gametes, egg and sperm cells, the alleles, different forms of a gene, for a trait separate so that each gamete carries only one allele for each trait. This law explains why a child might inherit a recessive trait that was not apparent in the parents, but was hidden as a recessive allele. The law of independent assortment further expands on this concept, stating that genes for different traits can segregate independently during the formation of gametes. This law is the cornerstone of genetic diversity, explaining how a child can possess a mixture of traits that might not necessarily resemble either parent in totality. Building on the foundation of Mendel's laws, the mechanisms of allele segregation and independent assortment come into play. Allele segregation is a critical process during meiosis, where each allele for a trait is separated into different gametes. This segregation ensures that offspring receive a random mix of alleles from their parents, contributing to genetic variation. Independent assortment further shuffles the genetic deck. It refers to how different pairs of alleles are passed to offspring independently of each other. This mechanism ensures that traits are mixed and matched in various combinations, leading to the unique genetic makeup of each individual. In conclusion, the exploration of Mendelian genetics through the lens of the 2A reinforcement and 6A conjoined triads reveals a fascinating world of genetic predictability and diversity. Mendel's laws of segregation and independent assortment lay the groundwork for understanding how traits are passed down through generations, contributing to the diversity of life that we observe. As we continue to unravel the complexities of genetics, the principles established by Mendel remain as relevant today as they were over a century ago, guiding us in our quest to understand the blueprint of life.